And talking about cooperation, collaboration, cross borders, things do overlap, don't they? Because our next session is on leading innovation. And it's one of the key words, isn't it? Innovation, collaboration, data sharing, cutting edge advances in therapies. I'd like to call on Dr. Patrick Couvreur, an emeritus professor at the University of Paris-Saclay, also a senior member of the University's Institute of France. Welcome.
Thank you very much for inviting me to deliver this talk on advanced nanomedicine for cancer treatment. Okay, so what is a nanomedicine? Just to remember you that a nanomedicine is in fact a drug which is encapsulated into a nanocarrier with a size in between a few 10 to a few hundred of nanometers. And as you can see here, if the encapsulation is well done, well, you will allow to protect the drug from the degradation. A typical example is with the messenger RNA. Also, because of the small size of those nanotechnologies, you can improve the intracellular penetration of drugs. And it is possible by chemistry to decorate those nanocarriers with specific ligands, which will allow a better cell targeting, and therefore a better therapeutic index. So the first technology that we have developed already a couple of years ago, as you can see, are the first biodegradable nanoparticles for human use based on polyalkyl cyanoacrylate. In fact, those nanoparticles are starting in terms of preparation by the anionic polymerization in a water medium of a cyanoacrylic monomer, which in fact is a biodegradable surgical glue. And so by emulsion polymerization process, this polymer is forming, as you can see here, nanoparticles with a size below or around 100 nanometers. And after intravenous administration, well, as you can see here in this autoradiographic picture, those nanocarriers, those nanoparticles will concentrate into the hepatic tissue. And this is the reason why we have used those nanoparticles for the encapsulation of an anti-cancer drug, doxorubicin, for the treatment of the multi-drug resistant hepatocarcinoma. As you know, in a lot, not all, but a lot of cancer cells, you have some efflux proteins that you can see here, which will induce the efflux of the anti-cancer compound from inside of the cell of the cancer cells to outside of the cancer cells. It is a way the cancer cells are using to fight against a chemotherapy. And we did here, just to illustrate that, a typical experiment where we took a human hepatocellular carcinoma, and if you are incubating increasing concentration of doxorubicin, you can see that all the cancer cells survive to the chemotherapy. And so the idea we had is to make the doxorubicin invisible for the recognition by the P-glycoprotein by encapsulating the doxorubicin into those nanoparticles. And you can see that it is working quite well, at least in vitro, because after incubation of doxorubicin as nanoparticles, now you can see that you are able to restore the sensitivity of those resistant cancer cells. And then, of course, we moved in vivo in preclinical cancer models. In general, we observed, of course, an increase of the survival because of this modification of the biodistribution. And a typical example here is the use of a model. It is a hepatocarcinoma in transgenic mice. And you can see that after administration of the doxorubicin loaded into those biodegradable nanoparticles, there was a huge increase of the apoptosis of the cancer cells comparatively to the treatment with the doxorubicin as a free drug. And this allowed us to create a startup company, Biolliance, which entered, in fact, the stock market in 2005. And this allowed us to start with a clinical trial. And the specialty name of those doxorubicin nanoparticles was the LivaTac. You can see here the result of the, excuse me, to go back. The result of the phase 2. You can see here in continued line that the survival rate of the LivaTac nanomedicine was increased comparatively after administration to those patients with resistant hepatocarcinoma, comparatively to the standard treatment, which was the chemombolization here in broken line. And this allowed us to start with a phase 3 clinical trial, which was a multicentric clinical assay in over 40 hospitals. And very rapidly, the LivaTac obtained the fast-track status from the FDA because the nanomedicine was well tolerated by the patients. But at the end, as you can see here in the survival curve, the LivaTac tended to show a similar survival curve than the control arms. Because in the meantime, in fact, the standard treatment has changed from the chemombolization to a multitherapy. And unfortunately, the nanoparticles, the nanomedicine, came too late and missed the opportunity to become the best in the class for this indication, and no nanomedicine could be put on the market. The mistake probably was that the startup company was willing, the people involved with the finance of the startup company, because entering the stock market, they were willing to do the third phase clinical trials alone without any collaboration with a big pharma. I think it was really a mistake. But in the meantime, from, I would say, more fundamental point of view, we have improved the technology. You can see here that we have functionalized those nanoparticles by click chemistry with biotin. Why? Because you know that a lot of cancer cells are hyper-expressing the biotin receptor. And just one example to illustrate that, we used MCF7 breast cancer. The cancer cells were labeled in green. And you can see that after incubation of the nanoparticles, which were biotinylated and labeled with a red fluorescent dye, the rhodamine, you can see that the cancer cells turned from green to a red, showing a molecular targeting ability. But during all this experience, we observed that finally, and this is true for all the encapsulation methodologies, that in fact the encapsulation on drugs in the current nanocarriers, including liposomes, nanoparticles, polymer micelles, etc., results always from physical encapsulation processes. This means, in fact, adsorption, entrapment, encapsulation. And this has, in fact, three very important limitations. The first one is a poor drug loading. This means, in weight, only a few percentage of the drug versus the weight of the transporter material, for instance, the polyalkyl cyanoacrylate polymer. The second limitation is a burst release, the uncontrolled burst release of the drug after intravenous administration. And this is due, as you can see here, in fact, to this fraction of the drug, which for thermodynamic reason is rather adsorbed at the surface of the nanotechnology than really encapsulated into its core. And therefore, the third limitation is that the drug release is not restricted to the target cells or tissues. And this is for that reason that we have moved from the physical to the chemical encapsulation paradigm. In other words, we did chemistry instead of physics. And to do that, we use a squalene because it is a natural and biocompatible lipid. And amazingly, this natural lipid adopts in water a molecular folded conformation, as you can see here. And we have taken advantage of this compact conformation of the squalene into water to link to the squalene used as a transporter material several drugs through a chemical link. And in fact, by a supramolecular chemistry, when you are putting those prodrugs, those bioconjugates into water, they will self-assemble as nanoparticles with a size of around 100 nanometers and with a much higher drug loading than after performing the encapsulation through a physical encapsulation process, because as you can see here, each molecule of squalane will be able to transport at least one molecule, one drug molecule. And also there is an absence of burst release because in blue you have a chemical link between the squalane and the drug. And if you are a good chemist, you can design a biocleavable linker which will be sensitive to an endogenous stimulus, allowing the drug liberation into the diseased cells or tissues. And this is really, I would say, a versatile and generic platform, which was applied, of course, to several anti-cancer compounds. I will speak about that, but also with some application in the field of the neuroscience for the treatment of intracellular resistant infectious disease. And you can also link the squalane, as you can see here to small interfering RNA with some application in the field of the cancer, and leading to nanoparticles with a drug loading which is over 90%. The first drug that we have encapsulated, that we have linked, in fact, to the squalane is the gemcitabine. When you are putting the gemcitabine squalane prodrug into water, by cryotransmission electron microscopy, you can see the appearance of those nanoparticles with columns, which correspond, in fact, to a supramolecular organization of those molecules, corresponding to an inverted hexagonal phase that we have discovered, demonstrated by X-ray diffraction at small angle scattering. And of course, we have tested those squalane gemcitabine nanoparticles on different preclinical models. One example here is the P388 leukemia. You can see that this cancer is absolutely resistant to gemcitabine, and that after the intravenous administration of the squalane gemcitabine nanoparticles, the tumor has completely disappeared. And to show you that, in fact, it is a very flexible concept, we have linked now to the squalane here the doxorubicin, and in that case you can see that we obtained elongated nanoparticles. And after intravenous administration, this allowed to have here in green much higher concentration of the squalane doxorubicin prodrug than after administration in red of the doxorubicin as a free drug with a very rapid blood clearance. And I will not enter into details, but after intravenous administration of the doxorubicin squalane nanoparticles, you will completely change the biodistribution of the drug, and especially you will dramatically decrease the concentration of the anti-cancer compound into the cardiac tissue. And you know, of course, that in the clinic it is a major limiting toxicity of the doxorubicin. It is the cardiac toxicity. And this allowed to have an increased anti-cancer activity. We tested that in a human pancreatic Mia-Paca cancer, also in a murine lung cancer. You can see that those experimental tumors are absolutely resistant to doxorubicin injected as a free molecule here in red, and that after administration of the doxorubicin squalane nanoparticles, you completely inhibit the tumor growth. And also we decrease the toxicity dramatically. You can see, of course, here in red that the administration of doxorubicin as a free drug is associated with a huge increase of the troponin level, which is a marker of the cardiac toxicity. And after taking some biopsies, you can see very clearly the vacuolization of the cardiomyocytes. And after administration of the doxorubicin squalane nanoparticles, at the same dosage in terms of doxorubicin, even the double of the triple, you can see that the level of the troponin was absolutely normal, and also the biopsies. Now, my last slide is to show you that it is also possible to design multifunctional nanoparticles by using metal oxide frameworks and doing nanoparticles with this material. In fact, metal oxide frameworks, as you can see here, are based on iron oxide clusters, which are complexed with diacids. And depending on the flexibility of the diacids, the molecular weight, you can design pores which are perfectly adapted to the anti-cancer molecule or molecule that you would like to encapsulate. And what is interesting is that the iron oxide clusters are decreasing the T2 signal at magnetic resonance imaging. So this allows you to do some imaging. And this was the concept we have developed in 2010 of nanotheranostics, corresponding to multifunctional nanocarriers, which are able to combine a therapeutic functionality because you have a drug encapsulation, but also with a diagnostic functionality because you are able to do imaging. And I would like to finish by acknowledgement of all the people of my group who collaborated more or less to one or another example I've shown to you, and also of the European Research Council for 2 grants that I have obtained for the development of this coagulation technology. Thank you very much for watching.
Thank you very much. The incredible world of nanomedicine. Thank you very much, if you'd like to take a seat. And last but not least, the last speaker in this very last session of the day, Leading Innovation, I'd like to welcome to the stage to talk about chemical control of cell adaptation, Dr. Rafael Rodriguez from the Curie Institute here in France. Welcome.
Thank you very much. Thank you also very much for welcoming me. It is a bit difficult to speak after Patrick. Patrick, I very much look up to you. If I can achieve, some of what you've done in my career, I'll be happy. So my lab focuses on cell adaptation, which, as you know, is the capacity of a cell to change and to adopt a new identity when exposed to environmental clues, which could be the tumor microenvironment, but which can also be the drugs that we use in the clinic. And you might know that Darwin said in the past, it's not the strongest nor the most intelligent to survive, it is the most adaptable to change. And we now know that there is a very strong contribution of non-genetic effect in the cell that allow cancer cells to adapt to the treatment we have. And so if we understand the chemistry underlying cell adaptation, only then can we develop a drug that might interfere with the process of metastatic dissemination and drug relapse. Right, so before I start, I'd like to acknowledge all the people I've been fortunate enough to work with over the years, my lab, and the great Juan Giovanna, somewhere, I cannot see him, Pascal Hamel, and Nelson. Thank you so very much. Without them, this work would not have been possible, and obviously the funding agencies, including Institut National du Cancer. Right, so as I said, cells can switch between different phenotypes, and if I want to be caricatural and to cut a long story very short, state 1 is a state that proliferates a lot, typically a state that will respond to cisplatin and irinotecan, paclitaxel, and 5-fluorouracil. And we've got the other state, the state that is more quiescent, and if it is quiescent, it doesn't respond to the drug targeting microtubule or the genome. What my lab has discovered is that this switch is made possible by 2 metals that are redox active, in particular copper and iron. And so today's is about iron. And I will try to explain to you how that works. This is a slide that recapitulates the past 13 years in my lab. And so here I've listed a few papers from my group. I cannot list them all, but if you want to have something that is coherent, you can start with 2017 and the one that was published last month, and there is a story that makes sense and that shows the evolution of our thinking in the lab. So we did stumble on that protein, the plasma membrane protein that is known as CD44, which you might know is coined as a cancer stem cell marker, which you would find being upregulated in residual tumors. You will find it to be upregulated in the metastatic population compared to the primary cells. This CD44 marker is associated to tumor CD capacity of a cell. And obviously it is associated to drug tolerance. So what was known about CD44 is that it is the main receptor of hyaluronic acid, and what we have discovered is that the hyaluronates that are in the stroma can interact with metal ions, in particular copper and iron, And once this complex is bound to CD44, CD44 will be endocytosed, taken up by endocytosis. This vesicle will mature. There will be acidification, which will release the metal, and then copper and iron can be distributed inside of a cell. We have discovered two things. The first is a new chemical reaction that is catalyzed by copper-2, not copper-1. Copper-1 was known in mitochondria, but not copper-2. Copper-2 activates hydrogen peroxide to oxidize NADH. NAD plus is the co-substrate of most of the Krebs cycle enzyme, and so this reaction now can be put next to the Krebs cycle because it fuels this enzyme and allows the production of ketobutyrate and acetyl-CoA, two metabolites that are required for acetylation and demethylation of histone in the nucleus, meaning that it is copper-2 in mitochondria that will increase when cells transform to allow a reprogrammation of the epigenetic landscape, which will allow transcriptional programming of cancer metastasis and drug tolerance. That is the first one. The other thing that we found is that there is an increase of nuclear iron. If you compare metastatic cells or drug-tolerant cells, if you isolate the nuclei from those populations to compare to the proliferating cells, you will find more iron in the nucleus. Why is that? In biology, we always coin metals as trace elements or cofactors. In chemistry, we consider these metals as catalysts. Catalysts, why? Because they can capture or release an electron. Here, iron 2 is the metal catalyst that allows histone demethylation. Without iron, there is no demethylation. Without demethylation, there is no upregulation of metastatic genes and genes involved in drug tolerance. Of course, once you know this, you can develop new drugs that will target copper-2 in mitochondria or target iron in the lysomal compartment, either to block plasticity or to induce cell death. Here, what we have done, thanks to Pascal, was to look at pancreatic ductal adenocarcinoma, whereby we isolated healthy tissues, or in this case, we just sorted the CD44 high-low population, and we could tell here that indeed there is more iron in the CD44 high population, which is quantified, and we compared this to the healthy tissues that were collected next to the tumor, such that you can see whether or not there is more iron in the cancer tissues compared to healthy ones. Next, there is a FACS experiment whereby we use a specific probe that detect reactive iron 2 in the lysomal compartment. And you will see that here in the CD44 high population, there is more reactive iron 2 than there is in the CD44 low population, which is coherent with the scheme I just showed. With this knowledge, being a chemist, what do you do? Is it possible to design a small molecule that will selectively accumulate in the lysomal compartment? That's characteristic number one we're looking for. And the second is this compound capable of chelating, interacting with the iron that is available to promote the redox activity of the metal and to promote oxidation of lipid membrane to induce death? And so we came up with this concept we called phentomycin. Phentomycin coined after the British chemist Henry Fenton in 1894, who discovered phenton chemistry. And so we synthesize this bifunctional compound, Fento-1, whereby you've got the red part and the green part. The red part accumulates as the plasma membrane. The green part target iron. After endocytosis, the green part ends up in the lumen of the lysomal compartment, is free to interact with iron 2, will form an iron-oxo complex with hydrogen peroxide, and at pH 5, which is the pH of the lysomal compartment, you can initiate this radical process whereby the iron-oxo will abstract hydrogen to promote a radical that will react with molecular oxygen, and this propagate within the membrane. And little by little, these lipids are being oxidized, the membrane dissociates, and cells are going to die of this newly coined cell death mechanism that is ferroptosis. That was the concept. So it took us 5 years to design this compound that you see here in red and in green, the phentomycin, Henry Fenton. The red part targets the membrane, the green part targets iron. This reactive catalyst is formed inside of the cell, not outside. Here you see colocalization of Fento and lysotracker. You see the red, that's the drug. Lysotracker is the lysomal marker, and the yellow here shows it goes where we said. Now, what we've done with the tissue samples, the biopsies that Pascal gave us, was to monitor lipid peroxidation, which we've done by quantitative lipidomics. And so here you see the red square here. This is the treatment by phentomycin. Now, when you co-treat with lipophilic antioxidant tocopherol, you no longer see this, meaning that this compound in the biopsy triggers lipid peroxidation. FACS experiment, we looked at this CD44 high population treated with phentomycin. You see we shift from 54 to 13% overnight. So here we just take the tumor, dissociate the tumor, soak the tumor with the compound over 12 hours, FACS it, and we compare non-treated to treated, and you will see that this population disappears. Now, when you treat with ferroptosis inhibitors or iron chelators or tocopherol, the radical trapping agent antioxidant, you see that you protect these cells from dying. So really, we can trigger ferroptosis in these drug-tolerant pro-metastatic cancer cells. Now that data we never published, so that's not published, but I think that's quite a spectacular data to my taste. Don't ask me why we didn't publish it, but I like it very much. Here, this is a metastasis of pancreatic ductal adenocarcinoma that is localized in the liver. And so we collect both tissues, healthy tissues, liver tissues, known to be iron-rich compared to others. And we compare the amount of iron in the tissue compared to the PDAC. And you see there is more iron in the PDAC than in the liver. RONOX-M stands for lysomal iron 2. And you will see that in the metastasis, there is more iron 2 than LC tissues, more CD44 than LC tissues. And now when you look at treatment, phentomycin triggers lipid peroxidation, which you can see with body PIC11 staining. Nothing is happening in LC tissues, but you see that there is lipid peroxidation in metastasis. Oh, nice. So we can selectively induce lipid peroxidation in the cancer tissues of the CD44 high, drug-tolerant persistent cancer cells, is PDAC, from a biopsy. Now, when you look at how many cells remain, whether these cells are dying or not, we compare untreated sample. There is no effect of phentomycin on those populations. But you see that the CD44 high population switched from 18% in untreated to 2% in treated. Now, that population is a population that you see being prevalent in the metastasis of PDAC. And this is a population leading to cancer relapse, for which today FOLFIRINOX has little effects, and gemcitabine for that matter. Now, those data were collected by Nelson and Juan. Thank you very much. Here we looked at our compound and how this compound is performing compared to irinotecan, oxaliplatin 5-fluorouracil. I'm not going through this all, but basically we see that not only the compound works better, but it gives a full response. And here, no matter how much you put more of oxaliplatin and irinotecan, at some point you reach saturation, and we presume that it is because the cells adapt and stop dividing and survive. We will see a similar effect in a 3D organoids. And now what we have done was to compare the compound just by itself. Phentomycin alone has little effect. 5-fluorouracil or gemcitabine or oxaliplatin or paclitaxel have little effect. But now when you mix both together, you see that there is a synergistic effect. Why? Because you target the two states at the same time, the one that proliferates and the one that does not proliferate but can potentially migrate and promote metastatic dissemination. Another two data, the first one acquired by Emmanuel Charaf and Christophe Ginestier, also in Marseille, same institute, and the second one, JSL in Belaker and Harvard Med. Here we looked at SUM-159, which is a triple-negative breast cancer cell line that is known to be CD44 high, extremely metastatic, and probably the most mesenchymal of the spectrum of triple-neg we can find. Here the experiment is very simple. You take a dish, you treat yourself with doxorubicin, which is the standard of care in this case, or a standard of care. You wait for 48 hours, you kill 99% of the cell, you change the medium, and you see how many cells remain and how many of those can seed a new tumor. Is there left any cells with tumor-initiating capacity, which are those that metastasize? And you see this profile. Now you do the same experiment, but at the same time, you incubate the cells with phentomycin 1, and you see that there are much fewer clones that grow, meaning that our compound eradicate cells in the tumor-initiating cell state, which also correlate with poor prognosis, metastasis, and drug tolerance. Now, the last one is an in vivo experiment. That's an immunocompetent model of triple-negative breast cancer, the 41. That model was model of lymph node metastasis. Why? Because triple-neg most often disseminate in the node first. But also, this is interesting because the microenvironment in the node is a reducing environment, and the capacity for a compound to induce ferroptosis in a reducing environment is very difficult. And so here we just wanted to challenge our compound, but also to see whether or not this compound was initiating ferroptosis that could be immunogenic. So here you see the black curve is the exponential cell proliferation of the tumor, but when you treat with phentomycin every other day, after 3 weeks or few days, you see that there is inhibition of growth. And here are the key messages that we've been learning over the years. CD44 is not a marker of anything. In fact, you see it in development and immunity as well. So if anything, it does mark transformation. When cells acquire a new identity, very often you will see upregulation of CD44. So it's not so much whether or not CD44 is expressed, it is the extent to which CD44 gets upregulated in the context of adaptation that matters here. Of course, CD44 mediate iron and copper uptake. Iron promote epigenetic programming of cell adaptation. There is no metastasis without iron. As a matter of fact, we have done those experiments. Lysosomal iron is druggable. It is a cell state that defines cell death modalities, and this is very important because you can try to induce apoptosis in cells in the mesenchymal state. It's not going to work. Why? Because all the biochemistry has changed. However, there is more iron. There are fewer or lower amount, I suppose, of a glutathione, and it's a lot easier to induce peroxidation of lipid than to induce this. Also, the lipid composition of the membrane in the mesenchymal state is different. There are more PFAS, more double bond, easier to oxidize. And finally, and to me this is the most important, something that I didn't say at the beginning, cancer is a genetic disease, of course, but metastasis is often a non-genetic effect. And I think it is well established now that drug tolerance and cancer metastasis can be the result of non-genetic event. As a matter of fact, it is metabolic and epigenetic reprogramming we are talking about here. So here we say that controlling cell adaptation confers therapeutic benefit. Linus Pauling in 49 and David Yu in 21 have shown that if you correct the genetic defect of sickle cell anemia, you correct sickle cell anemia, you correct the disease. Correcting the genetic defect corrects the disease. Here we show that correcting cell adaptation, which is non-genetic, also confers therapeutic benefit. And with this, I will thank you all so very much.
Thank you very much. Thank you very much. Please take a seat. Fascinating insights into the future. Maybe we can end the session just with a quick question to both of you. We're actually running late, so the discussion can continue during the cocktail afterwards. But the same question I began this morning with Dr. Urrutia, which is what excites you at the moment about tomorrow? What is the milestone? What is the breakthrough that you're looking for in the short term, midterm? What's exciting you at the moment?
I can answer only considering the field of the nanomedicine, of course, because, well, I think that the vaccination against the COVID was probably a revolution from the point of view of the nanomedicine, because you know that the messenger RNA had to be encapsulated into a lipid nanomedicine. With no nanomedicine, no vaccination. And this opened now a very important field of research, because you can replace, of course, the messenger RNA of the spike of the virus by a lot of small interfering RNA, which will allow, for instance, to inhibit the expression of an oncogene, of this type of protein, etc., etc. There is a lot of application. And you have always also a second field of interest with CRISPR-Cas9. Because, of course, you can make now some gene addition with CRISPR-Cas9, but you have to do it in the right cells. And to do that, you need also to do some targeting of this CRISPR-Cas9 system in the cells where you are willing to perform the genetic modification.
Okay. Lovely to have that vision as we come to the end of the day. Dr. Rodriguez, the same question to you. I was going to say, la même question. The same question to you, influenced by the French here. Yeah, what's your next milestone, the next breakthrough? What are you excited about?
Well, I don't have a short-term mission. The only one is I look forward to the day where people like Pascal and other clinicians will be able to put into practice what we do in the lab, and I just wonder every day, how can we collectively turn this around? And so I'd like to thank you, everyone, for participation.
Thank you very much. And this is one of the events that perhaps can lead to that. So thank you very much. A round of applause to our guests.